Good afternoon, all of you. So now we are going to discuss another important topic, that is insect resistant plants. Insect resistant plants. So by using genetic engineering technique or biotechnology techniques, how we will create genetically modified plant, that is insect resistant plant, we will see. See, come to previous class once. What are the advantages of genetically modified plants? So first advantage or application is genetically modified plants are more tolerant to abiotic stresses. Second advantage is it reduces reliance or dependence on chemical pesticides. And third one, increased efficiency of absorption of mineral nutrients by the plant body. And one more thing, it enhances nutrient quality. So that genetically modified plants prevents post-harvesting loss also. So this technique is also useful for the production of pest-resistant plants. It is also helps in production of insect-resistant plants. Right. So up to here we discussed in previous classes. Right. Then, so not only these uses, that genetically modified plants also act as alternative resources to the industries in the form of starch, in the form of fuels, in the form of biopharmaceuticals, right? So in addition to genetically modified plant advantages which we discussed in earlier classes, so that genetically modified plants also act as alternative resources to the industries in the form of carbohydrates, in the form of starches, in the form of fuels, in the form of biopharmaceuticals, right? So how the genetically modified plants, otherwise how genetic engineering technique, right, useful for the creation of insect resistant plants. So we will see one, right? See, BT toxins, BT toxins. So one type of poison or one type of toxin it is. That BT toxin produced by the one of the bacteria, that is Bacillus thuringiensis, right? What it is? BT toxin. So BT toxin, one of the poisonous substance, that poisonous substance produced by the one of the bacteria, important bacteria, that is Bacillus thuringiensis. Right? So this Bacillus thuringiensis produces one type of toxin or poison, that is BT toxin. So that BT toxin kills insects, right? That BT toxin kills insects, various insects. So that BT toxin zine, BT zine cloned in bacteria. See, by using genetic engineering techniques, that BT gene, what is BT gene? So Bacillus thuringiensis toxin encoding genetic material, right? So that BT toxin or poison producing genetic material isolated from that bacteria and cloned in bacteria, right? Isolated from the Bacillus thuringiensis bacteria and it cloned in other bacterial members and finally transferred to the plants. And finally, that BT genes transferred to the plants. So when we transfer that BT genes into the plants, what will happen? That plant modified into genetically modified plant, right? So that plant, whenever we transfer, so that BT genetic material into the plant cell, so automatically modified into transgenic plant or genetically modified plant, ZMP. So what is ZMP here? Genetically modified plant. Okay, right. That genetically modified plant act as biopesticide or bioinsecticide, right? So biopesticide, it acts as biopesticide or bioinsecticide. So what is the difference between pest and insects? See, all insects are pests, right? All insects are considered as pests. but all pests are not considered as insects, right? So here that BT genetically modified plants are act as biopesticides or bioinsecticides, right? What is the use of? See, they reduces the uses of insecticides or pesticides, right? So these plants reduces the uses of reduces the uses of pesticides and insecticides, right? So by using genetic engineering technique, easily we will create bioinsecticidal plants, biopesticidal plants, right? So for example, you can take BT cotton, then another one, BT brinjal, BT cotton, BT brinjal, BT rice, Bacillus thuringiensis rice one. So BT cotton, BT brinjal, BT rice, BT maize, and BT potato, Bacillus thuringiensis potato, and soya bean also. BT soya bean. So like that, by using genetic engineering technique or advanced techniques, easily we will create insect resistant plants like BT cotton, BT brinjal, BT rice, BT maize, BT potato, and soybean. Right? In that, we will take one plant, that is BT cotton. So how we will create BT cotton? So how it act as insecticidal plant, we will see now, right? So just brief introduction related to insect resistant plants here. See, BT toxin or poison, right, produced by the Bacillus thuringiensis bacteria. Bacillus thuringiensis bacterial members. So we isolate that bacterial gene or BT gene from that bacteria and insert it into the vector and cloned that vectors in bacteria, right? And finally, we transferred it into the another plants one, right? So automatically, the plants modified into genetically modified plants or transgenic plants. So that genetically modified plants or transgenic plants act as biopesticide or bioinsecticide, right? So why we call it as biopesticide or bioinsecticide? They are highly resistant to certain insects, certain pests. That's the reason we call them as biopesticides or bioinsecticides. So they reduces the usage of insecticides or pesticides. So best example, BT cotton, BT brinjal, BT rice, BT maize, BT potato, and BT soybeans. But nowadays we are using BT cotton, right? Well-known example, BT cotton. So how we will create BT cotton by using genetic engineering technique? We'll see step by step, right? Now, small side heading, BT cotton. So in BT cotton, BT stands for Bacillus thuringiensis. So what is the meaning of BT? Bacillus thuringiensis. Is it bacteria or virus? Bacteria. Bacillus thuringiensis, bacteria, right? So that BT genes or BT toxins or BT proteins produced by the some strains of Bacillus thuringiensis members. BT proteins. So BT proteins produced by some strains of Bacillus thuringiensis members. BT proteins produced by some strains of Bacillus thuringiensis members. Not all, some strains. Strains are nothing but types. Some strains of Bacillus thuringiensis. Bacillus thuringiensis members, right? So this Bacillus thuringiensis members, otherwise that BT protein encoding genetic material, BT protein encoding genetic material present in some strains of Bacillus thuringiensis members. So that Bacillus thuringiensis members producing Bt proteins are highly poisonous or toxins, right? So that Bt toxins existed or Bt toxins produced as crystals at a certain age or at a certain phase of the bacteria, right? So Bt toxins produced as crystals in the form of crystals at a particular phase of the bacteria, So these Bt toxins are highly poisonous, highly poisonous, right? So they kill certain insects. So what type of insects? They are, one is lepidopterans. Insect name it is lepidopterans. Then second one, dipterans. So lepidopterans, dipterans, and third one, coleopterans. Right. See that Bt toxin or Bt poison which is produced by the Bacillus thuringiensis members kills insects like lepidopterans, coleopterans, dipterans. For example, you can take lepidopterans. What type of insects comes under lepidopterans? So army budworms, tobacco budworms. So army budworms and tobacco budworms. So both of them comes under lepidopterans. Then come to dipterans. So in dipterans, flies and mosquitoes. So flies and mosquitoes comes under dipterans. And what about coleopterans? Beetles. So that beetles comes under coleopterans, right? It means that poison, Bt toxin or Bt poison produced in the form of crystals at a particular phase of the bacteria. So that protein crystals or Bt toxins kill certain insects. They are lepidopterans, coleopterans, dipterans. Lepidopterans are nothing but army budworms, tobacco budworms. Dipterans are flies and mosquitoes. Then coleopterans are beetles, right? But that inactive prototoxins or Bt toxins are inactive inside the bacterial cells. It means they do not cause any harm to the bacterial cell or plants. So they do not cause any harm to the bacterial cells or plants. Why? Because they are inactive. But when they converted into active states, we'll see. Just note down the points again, I'll explain you, right? Bt proteins produced by some strains of Bacillus thuringiensis members. That Bt toxins or Bt proteins produced as crystals are produced in the form of crystals at a particular phase of the bacteria, and they are highly poisonous to the insects. They kill certain insects. They are lepidopterans, coleopterans, dipterans. Within the brackets, that insect names also you have to write. All right. Is it endotoxin or exotoxin? Endotoxin. Why we consider it as endotoxin means, so the toxin produced inside the bacteria, hence it is called as endotoxin. The two we call it as protoxin. Why we call it as protoxin? It is inactive inside the bacterial cell. That's the reason we call it as toxin, first form of toxin, right? So continuation for this, BT cotton. Continuation of that information. So that endotoxin or endoprotoxin, protoxin, inactive inside the bacterial cells. Inactive. So inactive inside the bacteria. Inactive in bacteria. Inside the bacteria. Right? And that too, they do not cause any harm to the bacterial cells or plant cells. But when they converted into active states, right? So whenever plant ingests the plant tissue, otherwise whenever insect ingests the plant tissue, what will happen? So that prototoxins or BT toxins enters into the insect digestive system. Insect digestive system, right? So endoprotoxin is in inactive states inside the bacteria and inside the plant cells also. But when it is active, so it is converted into active states whenever insect ingests the BT toxins or BT proteins, right? So it is converted into inactive toxin, converted into active states inside the gut of insects, right? See, it is inactive, that inactive toxin converted into active state in gut of insects. What is gut? Gut is nothing but digestive system of insects. So why it is active? Why it is inactive inside the bacterial cells? Inside the bacterial cells, there is no alkaline pH, right? But whenever that inactive prototoxins enters into the digestive system of insect, so due to the presence of alkaline pH, that inactive toxins converted into active toxins. What is the reason? So due to the presence of alkaline pH, right? Due to the presence of alkaline pH in the gut of insects or digestive system of insect, that protoxin or inactive toxin converted into active state, right? So after activation of toxin, BT toxin, it closely associated to the epithelial cells or epithelial tissue of digestive system, right? So after activation, the toxin, activated toxin simply. After activation, that activated toxin closely associated to epithelial tissue. So epithelial tissue which is present in digestive system of insects, and it forms minute pore-like structures, right? So whenever that active toxins closely binds to the epithelial tissue which is present in digestive system of insect, so they forms minute pore-like structures. They forms minute pores, and it causes swelling and lysis of digestive system. Swelling and lysis of digestive system. And slowly that entire insect digestive system leads to ruptures, and automatically it leads to death, right? So slowly that insect leads to death. right? So in this way, the plant biopesticide, the plant act as biopesticide or bioinsecticide, right? So come to point by point, one by one. See that protoxins are present inside the bacterial cells of plant cells. That protoxins in general inactive, but whenever that insects ingest the plant tissue, ingest the plant tissue means whenever that insect takes that plant tissue as food material, automatically that Bt toxins or Bt proteins also enters into the digestive system of insects. So inside the digestive system of insects, that inactive prototoxins converted into active toxins. Active toxins. So due to the presence of alkaline pH. See that activated toxin closely associated to the epithelial tissues which are present in the digestive system and forms minute pore-like structures. Minute holes or pore-like structures. It causes swelling and lysis of entire digestive system. So slowly that insect leads to death. So in this way, so that plant act as bioinsecticide or biopesticide. Okay, sir, how we will create bioinsecticidal plants by using genetic engineering, right? So diagrammatically, I'll explain you one by one. Diagrammatically, I'll give you that insecticidal plant creation, right? Now come to diagrammatic representation of insecticidal plant or Bt cotton. See, first we need to select Bt genes or Bt genetic material from the Bacillus thuringiensis, right? So first we need to isolate genetic material. Which genetic material? Bacillus thuringiensis genetic material or Bt gene we isolated from the bacteria. Which bacteria? Bacillus thuringiensis members. And one more thing. So we need to select on bacterial cell for the vector or plasmid. So which type of bacterial cell? Agrobacterium tumefaciens. Agrobacterium tumefaciens. So agrobacterium tumefaciens it is. So simply you have to write A and T, AT. So agrobacterium tumefaciens bacterial cell we selected for this experiment, right? See, it has circular double-stranded DNA. It has xenophore. In addition to xenophore, it has small circular double-stranded DNA. We called it as plasmid. Plasmid. Okay, we isolated plasmid from the agrobacterium tumefaciens. Tumefaciens. So we called it as Ti plasmid. No, Ti plasmid. So what is the meaning of Ti plasmid here? Tumor inducing plasmid. Tumor inducing plasmid. So that tumor inducing plasmid has tDNA at a particular location, right? What it is? tDNA. So that tDNA only disease causing genetic material. tDNA. Okay, right. We isolated Bt genetic material from the Bacillus thuringiensis member and Ti plasmids from the Agrobacterium tumefaciens. Okay, right. Both bacterial genetic material Bt genes and Ti plasmid genetic material so treated with same type of restriction endonuclease enzymes, right? So both Ti plasmid and Bt genetic material treated with same type of restriction endonuclease enzymes or EN. See what will happen? It resulting in the formation of same type of sticky ends, right? So it resulting in the formation of same type of sticky end. So where exactly it cleaves? That REN cleaves at tDNA. At tDNA. Whenever that restriction endonuclease enzyme cut the Ti plasmid at tDNA, so that tDNA or disease causing genetic material completely inactivated. Okay, right? See, both Ti plasmid tDNA and Bt genetic material treated with same type of restriction endonuclease enzymes, right? So it resulting in the formation of same type of sticky ends. sticky ends, right? So it is BT-zine and it is TI plasmid. So that BT-zine or BT genetic material inserted into the TI plasmid at the place of tDNA. At the place of tDNA by using ligase enzyme. Am I right? By using ligase enzyme, that BT gene, BT genetic material inserted into the TI plasmid at the place of tDNA. So our tDNA already inactivated now. So yes, at the place of inactivated tDNA segment only, we inserted that BT gene by using ligase enzyme, right? It resulting in the formation of chimeric DNA or recombinant DNA. So we call it as chimeric DNA or recombinant DNA. See here, it is rDNA, or we call it as chimeric DNA. So that recombinant DNA or DNA or chimeric DNA consists of two types of genetic materials. One is TI plasmid genetic material and another one is bacterial BT genes, BT genetic material. Okay, right. That rDNA molecule again inserted into the bacterial cell cytoplasm. So by using calcium chloride treatment or any one of the method, that rDNA molecule inserted into the host cell cytoplasm, right? So which cell act as host cell here? Again, Agrobacterium tumefaciens only act as host cell, right? So same thing, AT, Agrobacterium tumefaciens. It acts as host cell. See, some bacterial cells positive, some bacterial cells are negative. So that some strains or some Agrobacterium tumefaciens bacterial cells not having any vectors. So that type of vectors we selected here as host cells. Host cells. So which bacteria it is? AT. AT means Agrobacterium tumefaciens. Agrobacterium tumefaciens. Okay, right? That rDNA which we created from the TI plasmid and BT genetic material inserted into host cell cytoplasm. So now we call it as genetically modified host cell, bacterial cell. Okay, right? So that host cell or bacterial cell infected to the plant cell again, injected into the plant cell. Imagine it is plant cell. So it transferred that host cells are Agrobacterium tumefaciens cell, genetically modified cell, transferred to the plant cell. Okay, right. What will happen in plant cell? That Agrobacterium tumefaciens bacteria transfers tDNA. At the place of tDNA, it has BT genetic material, right? At the place of inactivated tDNA, we inserted BT genetic material, right? Along with BT genetic material, the tDNA transferred to the plant cell cytoplasm. So that tDNA along with BT genetic material associated with plant genetic material, incorporated to the plant genetic material, right? So that tDNA along with BT gene, tDNA along with BT gene or BT genetic material incorporated to the plant genetic material, associated to the plant genetic material. And when we culture that plant cell, now we call it as transgenic plant cell or genetically modified plant cell. When we culture that genetically modified plant cell on the nutrient medium in vitro conditions, it resulting in the formation of entire plant body. Right? It means by using tissue culture techniques, when we culture that plant cell on the nutrient medium, it resulting in the formation of entire plant cell. Already we have some idea, each and every plant cell having ability to generate entire plant body. We call it as totipotency. Each and every plant cell has ability to generate entire plant. We call it as totipotent capacity or totipotency, right? So in the same way here also, when we culture that plant cell which contains BT genetic material on the nutrient medium, it resulting in the formation of entire plant body. Okay, right. In this plant body, each and every plant cell consists of Bt gene. Bt gene. You can take any plant cell, either it is leaf cell, stem cell, or root cells. Each and every plant cell consists of Bt genetic material. So that Bt genetic material undergoes transcription and translation in plant cell cytoplasm and forms Bt proteins. Bt proteins. So total plant body, cotton plant body modified into transgenic, right? So we call it as genetically modified plant. ZMP. What it is? Genetically modified plant. Otherwise, we call it as Bacillus thuringiensis cotton. Why we call it as Bacillus thuringiensis cotton? So each and every plant cell genetic material associated with bacterial genetic material. What it is bacterial genetic material? Bacillus thuringiensis genetic material. So that Bacillus thuringiensis genetic material undergoes transcription and translation and synthesizes Bt proteins in each and every plant cell cytoplasm. So whenever insects ingest the plant tissue, cotton plant tissue, what will happen? Along with cotton plant cells, that Bt genes or Bt proteins also enters into the digestive system. So that Bt proteins are inactive inside the plant cell, inactive inside the bacterial cell. But when they converted into active state, whenever that Bt toxins enters into the insect digestive system, so that inactive toxins converted into active toxins. So they are associated to the insect digestive system. It means epithelial tissues of insect digestive system and makes a small pore-like structures and causes swelling and lysis of entire digestive system. Slowly that insect leads to death. Right? So in this way, that cotton plant act as insecticidal plant, bioinsecticidal plant or biopesticidal plant. So what is the use of this bioinsecticidal or pesticidal plant? So it reduces the usage of agrochemicals, right? So it reduces the usage of chemicals, pesticides and insecticides, right? So that's the reason nowadays we are using Bt cotton plants instead of normal cotton plants, right? So how we'll create Bt cotton plant? Listen carefully. So this diagrammatic representation is important. So total concept is in that diagrammatic representation, right? See, first we have to collect Bt gene or Bt genetic material from the Bacillus thuringiensis bacteria, right? So we have to isolate Bt gene from the Bacillus thuringiensis bacteria. But how we will isolate? So already I told you in earlier classes, by treating with various enzymes, easily we will collect genetic material from the any bacteria, right? So first step, we isolated Bt gene or Bt genetic material from the bacteria, Bacillus thuringiensis bacteria. Second step, again, we have to select another bacteria, that is Agrobacterium tumefaciens. Agrobacterium tumefaciens. Right. So we have to isolate Ti plasmid from the Agrobacterium tumefaciens. Second step is we isolated Ti plasmid from the Agrobacterium tumefaciens. So why we selecting Ti plasmid song instead of PPR 322? Why? Because so for amplification of desirable genetic material in plants, always we're using Agrobacterium tumefaciens only. Why? Because within a short time, so it replicates rapidly inside the bacteria. Desirable genetic material replicates rapidly in Agrobacterium tumefaciens, and that bacteria can survive inside the plant cells. Right? That is the reason we selected Agrobacterium only. Okay, right? So the Ti plasmid has tDNA. tDNA, transfer DNA, otherwise tDNA simply. tDNA. tDNA is nothing but disease causing genetic material. tDNA is nothing but disease causing genetic material. Okay, right? See here, same type of restriction endonuclease enzymes we are using for fragmentation of Bt genetic material and tDNA. tDNA. Right, it resulting in the formation of sticky ends. What type of sticky ends? Same type of sticky ends. So, but when we treated the Ti plasmid with restriction endonuclease enzymes, the tDNA inactivated. Where exactly that restriction endonuclease enzyme cleaves TI plasmid at tDNA. So it means that disease causing genetic material completely inactivated. Okay, right. Again, we have to insert that BT gene into the TI plasmid at tDNA. At tDNA, it resulting in the formation of rDNA molecule, recombinant DNA or chimeric DNA. So that recombinant DNA or chimeric DNA inserted into host cell cytoplasm. Again here, agrobacterium tumefaciens only act as host cell. So already it has disease causing genetic material now, sir. Yes, but here two types of bacterial cells are there. So some bacterial cells not having any plasmids. So some bacterial cells have plasmids. So here we selected without plasmids, the bacterial cells without plasmids are host cells. Okay, right? We inserted recombinant DNA into host cell cytoplasm here. So now we call it as genetically modified bacteria. So that bacteria infected to the plant cell. Which plant cell? Cotton plant cell. All right. So that cotton plant cell infected with genetically modified agrobacterium tumefaciens. Right. So in this case, that host cell transfers tDNA with BT-gene. tDNA, it delivers tDNA with BT genes. Okay, right? That BT genes closely incorporated to the plant genetic material, associated with plant genetic material. Right. So that type of plant cell is considered as genetically modified plant cell. Why? Because why we call it a genetically modified plant cell? Along with plant genetic material, it has BT genetic material also, BT gene also, right? Bacterial genetic material also. Right. That. So when we culture the genetically modified cotton plant cell on the nutrient medium in vitro conditions or laboratory conditions, right, it resulting in the formation of new plant body, right? Already we have some idea. Each and every plant cell having ability to produce entire plant body. That condition is called totipotent capacity. Totipotent capacity. Okay, right. Due to totipotent capacity, that genetically modified single plant cell, right, gives entire plant body. Right. In that plant, each and every cell consists of BT gene. BT gene. So that BT gene undergoes transcription and translation process. It is nothing but gene expression that BT gene expresses inside the cotton plant and produces BT toxins or BT proteins. BT toxins or BT proteins. Okay, right? So in entire plant body, each and every cell consists of BT toxins or BT proteins. Right? That toxins or proteins are inactive inside the plant cell cytoplasm, inside the bacterial cell cytoplasm. They do not cause any damage or any disease to the bacterial cell or plant cell here. Right? But whenever insect ingests the plant tissue, along with plant cells, that BT proteins also enters into the digestive system of insects. Gut of insect means digestive system of insects. So after that, that inactive toxins converted into active toxins closely bunch to the epithelial tissue. That active toxins closely bunch to epithelial tissue of digestive system, insect digestive system, and forms minute pore-like structures. And it causes swelling and lysis means breakdown, right? Lysis of entire digestive system. Automatically, that organism, that insect, completely leads to death. That insects leads to death. So in this way, so that plant act as bioinsecticidal plant or biopesticidal plant, right? So when we will create genetically modified plant or BT cotton plant by using genetic engineering technique, uses of agrochemicals reduces, right? For reducing the uses of agrochemicals, we will create, right? We need to create that genetically modified plant. So how we will create? So by using this process of genetic engineering technique process. right? So nowadays we are using BT cotton plants, but we are not using BT brinjal, BT potato, but nowadays we are using BT cotton plants, right? Draw the diagram, flow chart. But it is insect specific. Insect specific, that BT genes are BT genes are insect specific. Insect specific. So that genes, BT genes varies from one plant to another plant, right? It depends on type of plant, type of target pest, right? For example, cry genes. Actually, BT genes called as cryzines. Why we call them as cryzines? They produces BT crystals. No, that's the reason that BT genes are considered as cryzine or cryzines. So that cryzines are species specific. For example, you can take cry1AC and cry2AB. Cry1AC and cry2AB. So these two types of cryzines or BT genes controls cotton bollworms. Cotton bollworms. So cotton bollworms controlled by the which genes, which BT genes? Cry1AC, cry2AB. Cry means crystals, BT crystals, BT toxins, right? So the cry1AC, cry2AB controls cotton bollworms. Then cry2AB, cry1AC, cry2AB controls cotton bollworms, and cry1AB controls corn borer. Popcorn, otherwise Zea maize. So Zea maize corn borer insects controlled by the cry1AB genes. So cry1AC 2AB genes controls cotton bollworms. Cry1AB controls corn borer. Right? It means that BT genes are insect specific. So that BT genes varies from target insect and plants. It depends upon target insects and plants. So that BT genes also considered as cryzines. So that cryzines varies from one plant to another plant, one insect to another insect. Insect specific, species specific, or insect specific. For example, you can take these two more. Cry1AC and cry2AB controls cotton bollworms. Cry1AB controls corn borer. So in this way, so that insecticidal plants are created by using genetic engineering technique, right? And remaining information we'll see in next class.